Hello everyone, I'm Daniel Klimmeck from EMBO Journal, editor there, and it's my pleasure to talk to Emmanuelle Passegue from Columbia University.
Thank you, Daniel.
In New York. And since we're at the Cold Spring Harbor Symposium for Senescence and Aging, we will focus on aging of tissue stem cells and their interaction with the niches during lifetime. So Emmanuelle, so you work at different tissues, but also including on the bone marrow and hematopoietic stem cells in different conditions. I also worked on the bone marrow, so I guess we agree this is a fascinating tissue.
Fascinating tissue, one of the most significant because it produces blood and it produces
we all need it, right? And
we all need it.
And specifically during aging, it is in the focus of both clinicians and basic researchers. So what has been the recent developments in that field? Maybe we start with that.
So first of all, just for the people listening, so you need blood production across the life range. So it starts during embryonic development, where you need certain type of blood cell in certain quantity, and then it's in the young adult, the peak of the reproduction age, and then in old individuals. So all these different phases of the timeline have different requirements. And that's where the importance of the regenerative engine of this blood system, the hematopoietic stem cell, which live within the bone marrow niche microenvironment and really tailor its output in terms of blood production. That's where the regulation of this cell becomes very critical. And we see fundamental difference in the way these hematopoietic stem cells are regulated during fetal development, in young adulthood, and what's the topic of this conversation, during the old age. So why so? Because this stem cell niche unit change over the timeline. And what is the best equilibrium in a young individual degrade over time, and you get to this niche which is now changed. A certain population of stroma cells which are lost, which are already gone, the bone integrity, the resilience of the bone is changed, and the marrow within it is affected by all of that. And what we have been studying, and a lot of labs are really studying that now, is this emergence of inflammaging, meaning the stroma cell are starting to secrete a lot of pro-inflammatory cytokine, which really have an impact on the hematopoietic stem cell and their downstream progeny, and the way blood production is being regulated. You're with me?
I'm still here. So that's interesting to me because intuitively you might say, well, inflammation is probably a bad thing to have, but at meetings like this one and over the last years, I think there's more and more evidence that it might actually be a good thing to, once in a while be exposed to inflammatory cues in order to train your stem cells so that they will know how to, they will not sleep there and sit there forever, but they keep being active. Is that the case for?
Inflammation was developed, the whole mechanism of inflammation and what you refer to as trained immunity has been built by nature to a reason, which is to make the body more responsive to sequential insult and injury. And the very important mechanism of response of the organism as a defense mechanism, which is, and that being studied, leave an imprint on the cell which have been exposed to inflammation. And a theory of aging associated with inflammation is that the accumulated load of all this inflammatory insult is a big part of what changed the biology of the old hematopoietic stem cell to make it from a very responsive and capable of producing the right type of cell needed at one given moment to a dysfunctional old cell.
So to the worst, basically, right?
Which basically doesn't make a lot of immune cell, B cell, T cell anymore, and it's very myeloid bias, myeloid cell being associated
the lineage balance is out of range.
Exactly, tissue degradation and inflammation. So yes, in this context, inflammation is a culprit of aging, is a bad actor. But it's not just a bad actor, because the way the cell responds to inflammation as well is by engaging stress response and resilience mechanism, which allows the stem cell to still be functioning in this change and flame condition. So we get to this trade-off, to this balance, so which is, yes, the environment change. Yes, the milieu become less welcoming and supportive, but the whole cell which are there are still making blood. If you look at the aging population, like the case of bone marrow failure or even of malignancy are
so luckily it's rare, basically.
Rare.
Yeah. So are you telling there's kind of a, I mean, the scientists, they talk about cell intrinsic and extrinsic factors and deficiencies of the niche or the stem cells. But is there, so in a sense, a learning curve of the hematopoietic stem cells? So they will, so because you emphasize there's kind of an imprint, but is there also a positive, you know, the aged HSC is wiser and kind of more capable of coping to a certain degree?
That's a little anthropomorphic.
I'm on the wrong track here, basically.
But and that's a very good question, in the sense that we know a lot about young, which is very young. Usually we take like 2 months old mice to model that, which is in human term kind of teenager stage, and we take usually very old, like 24 months old, 2-year-old mice, which is more like a geriatric 80-year-old individual. So what happened in between this young and old? Always this challenge, this inflammatory exposure, and all that shape the response and educate the stem cell and trigger some adaptive or contribute to cancer development or to the fitness emergence of certain clonal hematopoiesis disease. I hope I'm not losing people listening with all this jargon.
So that we need more data there, basically, better understanding.
Very important.
So that's
question.
one of the focuses of the field at this point.
Definitely one of the focus of the field, and the emergence of all the single-cell genomic approach and spatial transcriptomic, which allow to like really look at the niche cell
in vivo or in situ, basically.
In situ, in conjunction with the stem cell, is where there will be fundamental discovery.
So I guess everyone will agree in the field that we have a much more sophisticated and better databases for with in vivo data at single cell level to make our conclusions at this point. So, but you touched on the mouse models, right? And I guess we could is it like a lot of work is being done in mouse genetics and manipulating niche components, and but would you can these results be readily translated to the human patient? Is that a very analogous situation in the human niche, or would you say there's major differences which need to be considered?
So the difficulty of accessing the human niche is real. So just accessing human cell, so cord blood stem cell have been the
most accessible,
the tool of choice to study hematopoiesis in human. More and more studies now are moving to bone marrow HSC, comparing young and old as well. So we are really in the phase of
which is a heavy lifting in the lab, right?
A lot of data. The niche in human is very difficult because you need to have access to a non-inflamed, non-injured bone, which is really given for scientific purpose.
And like a bottom-up approach. So there's people I know in Switzerland. There's a group. They try to build a cellular niche for hematopoietic blood stem cells from scratch, basically. So they really is that a path to go?
Alternative which are being explored, like bone marrow on chip, derived from iPSC, so induced pluripotent stem cell, where you can regenerate the different element of a bone marrow, then assemble in like this fluidic platform to like reform scaffold, the organoid aspect where you use human mesenchymal stroma cell into like a sponge or bone structure and plant them subcutaneously in a mice to like make a pseudo-human bone. So all of that are possibilities.
New tools, right?
New tools which are used to start exploring this really fundamental crosstalk between the stem cell, their progeny, and the niche cell, and this bidirectional signaling, which clearly happened. You stem and progenitor educate some aspects of the niche regulation, and vice versa. The niche cell really educate.
I guess a lot of times we're getting at this chicken egg type of discussion, right? So is is the stem cell imposing a niche, and is it kind of higher up in the hierarchy, or vice versa, or both, or is it context dependently? I don't know.
I mean, there is clearly what's happening in situ in the bone, and there is what you can model when you take the stem cell out and transplant it in a different niche. I mean, we have done that by heterochronic transplantation or by heterochronic parabiosis, where you join together two mice and you have all stem cell coming from all hematopoietic stem cell coming up from an all mice lodging in a young bone marrow niche.
So you have a very clean setup.
No irradiation, no damage to the niche transfer. And so, and the whole stem cell, because they are activated in the sense that they have a lot of stress response mechanisms which are stimulated because they live in this old niche, which is not as nice and welcoming as the young niche. There is a lot of spillover. So a lot of this old cell naturally diffuse and engraft into the young bone marrow.
So they would you because
when you do that, they still stay old. So there is a component
something intrinsic
which is intrinsic to the old cell, which is not reverted by being exposed to a nice nurturing young niche.
And so that's interesting. So does it mean that there's a point of no return, where basically during aging, these features of an aged HSC are imprinted, and then it wouldn't be able to reverse to a more juvenile ability?
I mean, certainly by not by anything as subtle as changing circulating factor, like a lot of work has been done in the aging field with like this parabiosis, this systemic blood-borne factor, exercise, this kind of approach. No, these are not going to be really like affecting the whole cell. There is new approach. There is partial reprogramming, like going with transcription factor, pioneering transcription factor, which will change the chromatin landscape of the whole cell. Is that going to be reverting them to a more useful chromatin stage and therefore a better functioning activity? There is a lot of work as well in the metabolism of this whole cell, which is very different from a metabolism of a young cell. Again, because they have to adapt to a changed niche environment, and part of what makes a quiescent young stem cell is their ability to use glucose to go through the glycolytic process and really dampen oxidative phosphorylation, generation of ROS, and all of these trigger.
So it's really an orchestrated combination of the proliferative capacities, cell metabolism, and then surface factors expressed.
So that's why, if you think about how can we make an old stem cell better, it will be a multi-part strategy, because the niche will certainly be one element. If you can dampen inflammation in the niche, you are likely to like dampen a lot of this stress activation that or as well resilience mechanism that the stem cell is using. You can change the epigenetic. You can try to erase some of this accumulated effect of her life, well lived, basically, to reverse that. And that altogether might be helping to really produce a better tailored blood output to the system and prevent progression of clonal hematopoiesis or transformation to leukemia, which is really what
we are after, right?
What we are after. It's not so much to take the stem cell from an old person to transplant. That's not a clinical goal. A clinical goal is a better production of the cell needed, like a red blood cell for oxygenation, better response to vaccination, and preventing the degradation.
Quite practical, urgent needs of the patients. Well, Emmanuelle, thanks so much for your time and the insights. And
thank you.
We look forward to an update in a few, at the next meeting, I guess. So,
certainly.
Thank
you, Daniel.
Merci.
It was a pleasure.
Myself.